one thing I will say is it's an amazing turnout tonight here. I think we've had a lot of big turnouts for our short things this year. This might be the max. And hey, when you were back in high school and had to take sophomore biology, I think it was, how many of you can imagine that these many decades later, all you people would have turned out because you wanted to hear more about algae? I am guessing not a lot. But as we've all known for the last 5-10 years roughly, we have an algae. I like to call it brown crud, and some other friends do. Jan and others are calling it golden brown algae. And it has shown up every year. It seems like there's a few more reports that we get around the lakes of issues with golden brown algae. What's going on here? And Jan all along has been doing a lot of research, a lot of it for Torch Lake, but our environmental committee, Jan Garvey has been out with Jan and done sampling on Elk Lake as well. And Jan has been successful in ruling out some things, and he'll tell you where that research is headed right now. Tell you a little bit more about Jan. He is, without question, the authority on this issue. Do a Google search on Jan. It turns up headlines about his work on GBA, primarily with Torch. In the two Detroit papers, MLive, the AP, Michigan Public Radio, IPR, the local Record Eagle. Jan is recently officially retired from Michigan State University, where he co-directed the Center for Water Sciences. But I will attest that you can hardly tell he's retired. I've talked to him a number of times in advance of this, and we haven't talked about technical issues much. But every time I talk to him, he is looking through a microscope somewhere, and in his laboratory. They're here in East Lansing today, I think. And that's what retirement is like when you really love your work, I guess. He's been recognized for many of his projects. He's won many awards, published many scientific papers. We're lucky to have him working on this dreaded brown crud. That's all I wanted to say about that. So, Jan, good luck, and here we go.
It's a pleasure to be with you today and to talk about golden brown algae and many of the studies that we've done in Torch Lake, but now expanding out into other places so that we can learn actually what's going on in Torch and all the other places where this golden brown algae is growing on the bottom of the lake. So I'm going to talk for about 35 minutes and kind of get at this problem of what's causing this golden brown algae, or at least how we're approaching the problem and maybe why it's so difficult to figure out. So the golden brown algae problem seems to be something new. It's occurring in low nutrient lakes like Torch with very low phosphorus concentrations and low nitrogen concentrations. These are aerial photographs by Art Hoadley on the left-hand side here. In 2010, you can see that the sands of this bay are relatively white. And in 2020, this is exactly the same bay. I should have rotated the picture, but didn't. Actually, Becky Norris has asked me to rotate this several times, and so these waves in the bottom line up with these waves there to kind of document it's the same place. It is the same place, and golden brown algae has been occurring there for the last probably 8 years to 10 years at least. So if we look at the bottom of the lake, it looks like this in some cases, and it tends to take a lot of different forms, which is interesting. So I'm going to make you guys small over there. So the project's been going on for quite a while, actually. It started back in 2013 when we first had a meeting to kind of discuss the ideas. And in 2014 and 2015, we did some preliminary sampling of the benthic algae and exploring water chemistry, maybe even doing some experiments at that time. We expanded that work in 2016 considerably with a lot of sampling throughout the summer of water chemistry, groundwater, and the algae themselves, and running a few experiments. And this pretty much continued from 2017 to 2020 when we kept running different kinds of studies to see if we could figure out what it was. And actually ran a number of experiments last summer that I'll introduce you to at the end of the talk. And finally, in this year, we're transitioning. Well, we're still got lots of samples to analyze from previous work, but we're really working on linking the experiments to surveys both within Torch Lake and outside Torch Lake. So that's kind of a macro view of the project timeline. And I wanted to introduce the term benthic to you to make sure everybody understands what benthic means. Benthic are organisms that are attached to, but really not penetrating the bottom of aquatic ecosystems or even surfaces of any kind, really. It doesn't have to be the bottom. It could be pilings or docks, or it could be trees or and plants and wetlands. Anything attached to a surface in the water is referred to as benthic. And the species that are benthic, whether they're algae, invertebrates, fish, they're all different than the benthic species are different than the planktonic species. So a very different habitat in the water column. So you can look up a lot more research and details about things that have than I'll be able to talk about today. A lot of that is on the Torch Lake website, and I'll give you that information at the end of the talk. But today, what I'd like to talk about is I want to first describe what GBA is in its kind of worst, thickest case, if that's worst, and then describe the hypotheses that we generated pretty much at the very beginning of the study. And we've added some new hypotheses as we've gone along, but that drove that helped us target what to study to begin with. So I'll describe then some of the key findings over the first 6 years, and then describe a new hypothesis about how low phosphorus might be causing the golden brown algae. And I'll introduce a lesson from the Everglades that both describes where I've worked for 28 years on research in the Everglades, and it provides an example of how to approach problems, environmental problems, when you're trying to figure out what change in habitat has caused a change in something that you're interested in. So I'm going to take that diversion, and then that's going to explain what we're doing now. That's going to help explain what we're doing now. So that trip to the Everglades is, I think, worth the trip. So first, I want to acknowledge Three Lakes for funding a lot of the water chemistry analyses at my expenses and my time, and being such great hosts when I've been around the lake. And in particular, I want to acknowledge Dean Branson for all of his energy at the start of this project. And he's actually the person that called me up and got me involved with the project. And Becky has led the Water Quality Committee for many, many years. And then there's a whole host of people from 3 lakes that are just tireless volunteers in the field, in the air, all over to help out with sampling, and sampling when I'm not there. Often I just get samples being sent to me. There are other collaborators too from the U of M Biological Station, the water chemists there, Tim Vavrika helped out, and then 2 of my good friends, algae people, Rex Lau and Pat Koselleck, some people from Michigan State, and just a whole host of people helped out. So they, of course, need to be acknowledged in this project. So to introduce you to the golden brown algae, this is a piece of mat. So the mats are about a half an inch thick when they're really thick, and they're cohesive, so you can pick them up in your hand. And this is a picture of a mat up in the upper left-hand corner where I've just laid the mat down in a plate, and the surface pieces have kind of fallen off of some loose gray material at the bottom. So this is calcium carbonate precipitate when you look at it very closely under a microscope. But a lot of the calcium carbonate precipitate is anchored into this overlying golden brown crust. So the golden brown crust, if we stick it under a microscope and we focus in on it, is composed of these spherical cyanobacteria that produce mucilages that have golden brown pigments in them. In addition to these cyanobacteria, which are not toxic, known to be toxin-forming cyanobacteria, known to be, there are lots of diatoms. So diatoms are larger algae when looked at the same magnification as the cyanobacteria. This is a raft of them together. The picture down in the lower left-hand corner is an increased view, and diatoms are actually one of the dominant algae in the world and produce 25% of the world's oxygen. These microscopic organisms, typically just single cells, sometimes connected together in colonies, but still single cells with no multicellular organization into tissues. They have glass cell walls instead of cellulose cell walls, and they have golden brown pigments in them. So often the golden brown color of the algae on the bottom is just due to diatoms and not due to the cyanobacteria. There's differences in different mats. So this top crust is composed of a lot of mucilage-bound algae holding together, and those mucilages then penetrate down into probably a quarter of an inch of calcium carbonate precipitate that's all aggregated together. And I'm showing you these pictures of the calcium carbonate precipitate down here in the middle of the bottom row at successively larger magnifications. So you can see that it's white with golden brown dots in it, and those golden brown dots are diatoms, and maybe a few threads of cyanobacteria. So this is the structure of that mat. There's a lot of mucilage really tightly holding together the surface. That mucilage penetrates down into calcareous material underneath it, and that sits then on even a looser layer of calcium carbonate precipitates in different forms of calcium carbonate, maybe some calcium phosphates and some other things underneath that. So this is the reason I've shown this picture over on the right-hand side is this is one of the kind of key taxa that develops when the mats are formed most greatly at the end of the season. And I'll tell you its name later. So when that gang of us were together at the beginning, we came up with a whole list of hypotheses that we wanted to think about. So we were thinking maybe dracaenid mussels, like in the Great Lakes, are enriching the sediments and it's causing algae to grow. Maybe light conditions have changed and it's causing more algae or runoff, enriching the surface water and then causing algae on the bottom to grow. maybe we lost organisms that were disrupting or eating algae on the bottom. Maybe there was a non-native algae coming in. One of the things I was concerned about is that algae for biofuels and algal blooms that were causing harmful algal blooms were being reported a lot in the news. So I was concerned that because algae is in a lot of habitats, that people had just started noticing the algae on the bottom of the lakes and were just started to get worried about it. So it was really important to have those before and after photos by Art Hoadley and the observations that people around the lake have made. Thought about climate change in a warmer lake and then groundwater contamination. And I'll come back to this key question later. So as Bob said, I've worked with benthic algae and algae for almost 50 years on research. And with Rex Lau and Pat Kaselik and the knowledge of the people from around the lake, we kind of triaged these hypotheses. And it made the most sense that maybe septic waste or other kinds of groundwater contamination from fertilizers, either from ag or riparian lawns, that groundwater contamination was stimulating the algae on the bottom versus surface water. Just made more sense that it was coming from the bottom. Surface water should stimulate the algae in the water column, and the algae in the water column have not been increasing. Water clarity has not been decreasing to show that there's been more algae. So this groundwater contamination seemed like the place to start. So we designed our first early studies around groundwater contamination. And we also learned very on, based on the concentrations of phosphorus and nitrogen and some experiments that Rex Lau did and one of his students, that phosphorus was more limiting to algal growth than nitrogen. So we targeted phosphorus as the key parameter. And the approach that we took is, although we didn't sample all these locations, we sampled quite a number of these locations for groundwater. And we looked at benthic algae. And we've done other surveys looking at land use and the aerial photographs to come up with some characterization of how much algae is at different locations around the lake. And can we relate that to groundwater chemistry that we measured, or differences in land use that would indicate different levels of contamination of the groundwater? And although using visual assessments from GoPro cameras and the aerial photographs that Art Hoadley took, although there was variability among sites both in June and August, there was really no relationship between where we were measuring high phosphorus or kinds of land use, where there were a lot of houses and not a lot of houses, and where the GBA was occurring. So that the lack of that relationship does not support our hypothesis that it's groundwater contamination that's causing the problem. There were sites, for example, this one site, both in June and August, where there was relatively low GBA. So there's a little bit of consistency on where we find low and where we find high GBA. But there's just not a strong relationship between where it is and where we'd expect phosphorus contamination or where we measured it in the groundwater. The other major line of evidence that does not support the groundwater source of nutrients stimulating GBA is that there was a study done around 2005 by a group of people from Torch Lake, authored first by Norton Bretz, but Dean and Tim and Paul Roush were also associated with it. And they sampled groundwater. They did an extensive study of groundwater around Torch Lake. And when we compared the concentrations that they found in the 2006 report to the measured phosphorus that we saw from 2015 to 2020, they were about the same. There was not a difference. There was not an increase. So over the time that GBA developed in the lake, where it was, we did not see an increase in phosphorus in the groundwater. So again, we kind of have to look someplace else for a hypothesis to think about what is causing this problem. And so we've still got that whole list of hypotheses, but we're not finding good support for groundwater contamination being the reason. So we're looking at, we're not dismissing it, but we're looking other places. In fact, there are a lot of patterns that still make me think groundwater's got to play a role here somehow, but it does not seem to be the ultimate driver of the problem. So we go back to this question about what's changed in the lakes that could possibly be causing GBA. And while I was writing a report, I got into some of the data that has been collected by the Cooperative Lakes Management Program and the tip of the mitt over long periods of time, here from 2004 to 2020. And in all of the lakes, Clam, Bel Air, and Torch in the three lakes system, they all had a decrease in phosphorus concentrations over that time period. And diving into that data more thoroughly, so looking at, I think, I can't see the bottom of the screen here because of little Q boxes up on the bottom from Zoom, but I think there are 23 different lake locations that I downloaded data from tip of the mitt, and I also looked at cooperative lake management data, and consistently there were phosphorus decreases over these time periods in the lakes, in the larger oligotrophic lakes throughout this region. And it turns out that there's other evidence that I'm not going to talk about today, that this decrease in phosphorus might extend back all the way to the 1940s versus just being recent. So the time of this change in the environment, a decrease in phosphorus, the time of this change in the environment does correspond to the time that GBA was observed for the first time. So it gives you a lot of pause about, so what could, how can a decrease in phosphorus cause more algae? Well, it turns out that's actually really plausible. That there are two examples of low phosphorus causing algae to be able to grow. One of them is Didymosphaenia, which is being observed more and more in streams. In fact, Becky Norris has found it in the Rapid River, right? I've got that right. And so locally, it's being found. It's not Didymo that's causing the problem in the lakes, but Didymosphaenia occurs in low nutrient streams, and it cannot occur when phosphorus goes up. The Didymosphaenia disappears. There are hypotheses for why that occurs. So why doesn't more food for the algae make more algae? And it might be related to disease and other kinds of factors that might be affecting the didymo, that as phosphorus increases, it allows disease or something else to happen that kills off the Didymosphaenia. The other example is in the Everglades. So I'm going to now move into this Everglades example and help us think about how to approach the problem. So in the Everglades, there is a calcareous mat that develops on the plants underwater in this tannin-stained water. And then as the season goes on, that calcareous algal mat floats up to the surface. And it has sufficient structural integrity that it intertwines with the plants, and when it rains or wind blows, it stays on the surface. It has sufficient structural integrity that snakes crawl across the top of it and don't break it up. We can pick it up in cookie sheets and then cut it up into pieces and run experiments on it. So this is a calcareous algomat in the Everglades. And the same species that we observe in Torch Lake and in GBA is actually named after the Everglades. It's unique to the Everglades, but we're finding it in other places. It's called Encyanema everglanianum. This really important species in the GBA is Encyanema everglanianum. It's the same species. So these coincidences are not being lost on me. So it's possible that the GBA in Torch Lake is due to low phosphorus based on this work that we've done in the Everglades. So the region that I'm talking about is this water conservation area right here, this marshland here. This whole region is the Everglades. But we conducted a lot of research in this one area, this kind of trapezoidal, weird shaped area that has a canal on the north side and water from the agricultural areas enter that canal and produce a phosphorus gradient from north to south, with the north being contaminated and the south being very clean. So I was involved with research there to help the state of Florida and actually a bunch of lawsuits were going on about what was needed to control the phosphorus problem. And so I worked with Kurt Richardson here on the right, who was at the Duke Wetland Center, and he asked me to come down and work on the algae in this broader project. And we studied this phosphorus gradient in the Everglades, in the marsh of the Everglades. And one of the first things we did was address the question of, is it possible that an increase in phosphorus could cause the loss of this calcareous algomat? Because that's what was happening along this gradient. Close to the source of pollution, there was no calcareous algae. And when we would take the airboat from the northeast corner down to a site where we built, actually the Duke Wetland Center, not me, built these mesocosm systems out in the middle of the Everglades, we would see no calcareous algae, and then all of a sudden, we'd see calcareous algae growing in the open water areas of the marshes. So, it's just completely counterintuitive to somebody in 1995 to be going down there and think everybody's talking about the phosphorus pollution is causing a loss of algae, and this, it just made no sense to me. It was backwards. I was actually fighting that hypothesis. And fortunately, the Duke group was conducting this experiment, and in these channels where they released phosphorus out of these big tubs at the head of a channel, flow moved from north to south through these channels, and they would put phosphorus in very specific amounts in these tubs and then flush it down through these channels. So they created an experiment, and they basically asked the question, if we add only phosphorus, do these calcareous algomats disappear? And they did. They disappeared. You add the phosphorus and they disappeared. This is a low phosphorus habitat where phosphorus limits the growth rates of the algae. But we then found out that the reason that we lose the calcareous algomat is that the mat does not deposit calcium carbonate. The algae don't deposit calcium carbonate at high phosphorus. And that weakens the structure of the mat so that it can't resist the wind and the rain disturbance as it floats on the top. So it breaks up and it settles down to the bottom. So we understand basically from these experiments that phosphorus did cause that problem. And we used aerial surveys of these sloughs, looking down and taking pictures, and we found that we actually found this break point along the phosphorus gradient So this is 6 kilometers from that canal discharging agricultural water into the marshes. This is 7.76 kilometers, and statistically, there's a change in how much of the slough surface, which we were photographing, how much of the slough surface was covered by floating calcareous algal mats. And close to the agricultural water source, there was none. Almost always there were a few times that there were calcareous algal mats, but statistically at this 7.6 kilometers, all of a sudden you could almost always rely on almost 75% of the surface of the water was covered by calcareous algae. So on average, it was 75% of the surface, sometimes 100%, sometimes not so much. Something else was limiting them there, or something, maybe phosphorus was higher there for one reason or another. So we then use this data. We use this pattern on where along this gradient do we get these natural calcareous algal mats in the Everglades that are really an important ecological feature from both an aesthetic perspective. Here, people like calcareous algae, and people use the Everglades a lot for recreation, airboating and frogging and fishing and things like that. So they like that floating calcareous algal mat. That's what they're used, and that's what they grew up seeing. So we can use that relationship and what phosphorus concentrations and how they decrease along the gradient. We use that information to set a phosphorus management target for the Everglades at 10 micrograms per liter. So around 8 kilometers from that source of phosphorus, we had 10 micrograms per liter, and that allowed these calcareous algal mats to form. And that was turned into a regulation by the state of Florida and supported by the US EPA to protect the Everglades from phosphorus pollution. So it turns out that the relevance of this lesson is first in how do you approach solving the problem. And we've been doing this throughout the whole project. But now we're scaling up a little bit because the mats in the Everglades really are a lot like they're not exactly the same, but they're a lot like the mats in Torch Lake and other lakes that I've sampled that have this calcareous algal mat. So it's possible that a decrease in phosphorus is causing this problem. It's possible. We don't know that's the cause. It's possible. And why did phosphorus decrease? Well, we know Dracaena mussels, quaggas, and zebras can decrease phosphorus concentration in the lakes, and they have over the last 15 years or so. It's also possible that nitrogen deposition might be responsible for phosphorus decreases over a much longer time period, which will go along with some of the evidence that it occurs. The phosphorus has been decreasing for almost 80 years. It could be the phosphorus detergent ban, could be a change in phosphorus fertilizers, could be a number of things. But a year or so ago, and still, as dissatisfying as it is, I have other projects to work on, so I'm not trying to make work. I could not tell you that you should solve your groundwater phosphorus problems because I'm not at all convinced that's what's causing it. And I want to be as positive here as I was in the Everglades when I could say that it was phosphorus, the loss of phosphorus in the Everglades that was causing the loss of that calcareous algal mat. So here we're thinking about a reverse situation. But again, none of the hypotheses are off the table. So we continue to gather observations, and we're running experiments now. We're doing larger scale surveys, and we're collaborating with the USGS. So in brief, this is what that's about for last year and this year. So we're running experiments over on the right-hand side. Fred and Art have helped with these two, with deploying this, and Becky and other people helped with constructing these things. But these are two different devices. One is a frame that we put on the lake bottom. And the nice thing about using this versus other approaches that have been used in the past is that the frame could just be put on the lake bottom and it doesn't disturb the algae there. And in the square of the frame, we put a gelatin-like substance, agar, and kind of like slow-release time capsules that you take in medications. You can put nutrients, nitrogen and phosphorus, in that agar, and it slowly leaks out of the agar. So we loaded these arms with nutrients, and running a pilot in Lake Michigan, I saw really strong responses of actually Cladophora, the filamentous green algae Cladophora, to the nutrients coming out of the square frame here and leaking into the middle and around the edges. So we conducted an experiment where we didn't add anything to the frames, where we added just phosphorus, where we added just nitrogen, and then we added nitrogen and phosphorus both. And the color coding on this corner would tell you which treatment it was. And this is as we were setting them, because when we got back, there's filter cloth, there's a filter fabric that's wrapped around and zip tied to the PVC tubes filled with agar and some nutrient treatment. These were covered with algae growing on them. So it'll be really interesting and I'm actually working up the samples now, so I don't have any results to really describe, except to show that there is a big difference between the control, just visually. If you look at the filters from the controls and the filters from the nitrogen and phosphorus and enrichment, there's a lot more algae. But just because there's more algae does not mean that it's the GBA. Because the trick with low phosphorus algae is they grow slowly. They grow slowly, and because of the calcium carbonate and the mucilages that bind them together, and you get more mucilage with lower phosphorus, you get more goo and more calcium carbonate kind of anchoring this assemblage together, so they're resistant against waves or fish movements or invertebrates eating them. So they persist. So they just slowly grow over the summer, maybe even over winter, and then seed shallow water areas. So it's kind of this complicated ecology, but just think the calcareous algae are the tortoise versus the hare. They're the slow-growing organisms that eventually accumulate the high biomasses. So that's what we're looking for. We're not looking for just a lot of algae. We're looking for a lot of the algae of the right kind, the kind that we find in the GBA in these experiments. The other kind of experiment we ran was in bowls, and we added the agar to the bottom of the bowl with some sand on top, and we were just I ran two for a couple different reasons that I won't go into here, but we ran two to be as thorough as we could in running the experiments. So we're running Everglades-like experiments to find out if we add phosphorus, do we lose GBA? If we add phosphorus, do we get GBA? Do we have to add nitrogen too? If we add nitrogen, do we get more GBA because it depletes the phosphorus in some way? So we have results that we expect to see that would help us test some of these hypotheses about whether it's an increase or a decrease in phosphorus? Or is it potentially a change in nitrogen that's causing this problem? And then we'll relate those results, as well as the last 6 years of results in Torch Lake, to 2 surveys that we're running over pretty much a 3-year time period. We're running a 20 lake survey around Torch Lake to take advantage of the variability in the GBA in Torch Lake and see if we gather more information about groundwater and land use, which is where our USGS partners come in. Can we develop a relationship between where GBA occurs and groundwater or land use and potentially other factors that might be causing that GBA? In the same way, to increase the range of environmental conditions that we're working with, instead of just in Torch Lake or Lake Bel Air too, where we've done quite a bit of sampling, we're expanding the scale and sampling basically the entire northwest region of and actually down potentially Houghton Lake and Higgins Lake as well, where I've done work in the past, to look at the algae on the bottom of the lake, look at lake chemistry being collected by the Cooperative Lake Management Program and Tip of the Mitt, and see where do we have GBA, where don't we have GBA, where do we have the right species for GBA, where don't we have the right species for GBA, and try to understand along longer environmental gradients, where does the GBA occur. So finally, it's been really valuable, or we hope it'll be really valuable. We expect it to be really valuable to develop a relationship with another research team that can do something that we can't do extensively. It's just beyond our capacity to do. As an individual lake association, a bunch of volunteers and one research scientist helping out. And so we will continue, the Three Lakes Association and I will continue trying to understand the relationship between water chemistry and potentially herbivores and bioturbation is perturbation, disturbance of the bottom by organisms, the bio part. We're going to look at how surface water, interface water, groundwater, the water chemistry of the lakes basically affects the algae in the lake using all these different approaches that I've talked about and others that might be necessary. And then the next question, though, is what's caused those changes in chemistry, or what's caused those changes in organisms in the habitat that are causing GBA? And what natural factors could be important? Hydrology, geology, there's some signal, just these indicators that it might be some natural factors that mitigate whether we have GBA or not. But it's almost certain that something human related has caused this GBA to occur. and so we're trying, and that's where the USGS is going to come in. They have that expertise to tell us how natural factors and human factors are affecting water chemistry and may have affected invasive species in a way that the Three Lakes Association and I can say that those were the factors that caused the development of GBA in the lakes. And then that will lead us to, and without that, we don't know which way to go, that will lead us to a solution for how to solve the problem of GBA in the lakes, if it's practical to do that. Now, as I mentioned, there's quite a bit of resources and work that we've done over the last number of years and I wish I did have an answer for you so I could go off and do other things. But but if you want to read more about the work that's been done, is a lot of the early work is on the Three Lakes Association website. There's a report that I did in 2021 that I could not find on the website, but I know they've got it, and we can make that available on the website, I think, without too much trouble. And then I have just a gee whiz YouTube video that's about 15 minutes long, that if you want to go to that, it reiterates some of the early part of this presentation, and it would allow you to review it if you want to. So with that, I could take some questions and we could have a discussion. The question was from Don. Did your efforts in the Everglades reverse things so that algae was added, and you think the Torch Lake is reversible? No, it did not. So the reason that no, it did not allow restoration of the Everglades. When phosphorus contaminates sediments, particularly in wetlands, it takes a long time for that phosphorus to get buried. So those areas of the Everglades that are contaminated with phosphorus are contaminated for a long time. So, but what it did do is it provided a management target, and they now regulate unpolluted, all the polluted areas. They're trying to get it down to 10, so that eventually, after a long time, those wetlands will again support the calcareous algal mats and all the other things that there were. I mean, there were changes in macrophytes and invertebrates and many cascading effects through the whole ecosystem caused by the phosphorus pollution. Relating this to the northern Michigan lakes, Torch being the one that we're doing the most work in, it depends. So first, it depends if it's phosphorus. Let's run with that scenario. If it's phosphorus, if it's low phosphorus, if it's lower phosphorus, if phosphorus comes up a little bit due to two factors. One is dracaenid mussels have kind of reached their capacity, and I would guess they've kind of leveled off and they're kind of decreasing down. So and the other was nitrogen deposition. If you partner those two things, so I'm just running a scenario. So if it's phosphorus, again, if it's phosphorus, and you run a scenario then about what USGS is going to help us understand much more, put nitrogen deposition and zebra mussels have a zebra mussel dracaenid or quagga kicker on the end of this that just pop phosphorus down even more. If you if that's the case, and zebra mussels are not having the impact they did before, nitrogen deposition has largely changed. That means the landscape is going to contribute the phosphorus that it's historically contributed, and you'll come back to kind of the same place. And there's some indication, not enough to be certain, that phosphorus is coming back up a bit in the lakes. But again, there's just not enough information about that certainty. But if you go with that scenario, the phosphorus will gradually come up. And if we understand the mechanisms of why these calcareous algal mats are so successful, the phosphorus coming up, well, that will change their physiology so they'll no longer precipitate the calcium carbonate, which binds together with the mucilages. And so severely, the low phosphorus that so severely limits their growth that they produce a lot of mucilage, that will change their physiology in a way that they won't be able to produce the mats on the bottom. So that's kind of that's a good news scenario if you back it all the way out to those causes of the decreasing phosphorus and the low phosphorus causing the GBA. If, however, it is groundwater contamination by septic systems, it'll take a long time to solve that problem. Septic, going on sewers is not going to change your groundwater chemistry for many, many years until the nutrients that you put into the ground have dispersed. So any questions from the audience, or are you typing in questions from?
Did you hear that all right,
Have
Jen?
pH levels changed, and what would be the effect?
There is some, there's one hypothesis that yes, that it's the pH, but the lakes have, so I don't think the pH would change the species of algae. I don't think the pH could have changed across a range that would cause that shift in species. It's just my experience working with algae. So it's not that, I don't consider that mechanism highly likely. Could it have, could the lakes become harder, and could the change in pH be caused by more calcium in the water? And that's moderately plausible. But I'm guessing there's enough calcium in the water. So this would also be something where we get involved with some biogeochemists but we have been involved with them, and none of them has really said as the lake pH that have postulated that the lake pH could have changed that much that we now have calcium carbonate deposition and we didn't have it before. So so that's how, that's an example that I'm listening to all these hypotheses, and everybody's listening to them. And I'd encourage you not to throw, and I'm not dismissing the hypotheses, but I'm trying to prioritize which ones we should address, which ones we should address next with the limited resources and the step-by-step process that we're going through.
Have we ever looked at historic water temperatures and high temperature levels in summer, that effect on algae?
That does, and again, that's plausible. But over the time period, so I had one of my students was amazing using satellite imagery, and he used satellite imagery for Torch Lake for I think 25 years, something like that. I've got that figure someplace buried in some of the reports. And over the time period that the GBA developed, there was no change in lake temperatures. Maximums, seasonality of it, we could not detect a pattern of change associated with that. So for that reason, it's not high on the priorities, not gone. And it could be, I mean, it's not just, it's also not necessarily just one factor. And maybe it was one thing before and now it's something else. So, yeah, so, and the kinds of changes in the algae, I'm not thinking it's a temperature related change, but again, it's plausible. It's plausible and not off the list of possibilities, and one that we've considered. I'm going to go to one of the questions on the chat, if it's okay.
Just
so Richard asked.
let me ask you this as follow up to that, just to tweak what the question that was asked. What about ice cover? There we have more winters, especially on the big deep lakes like Elk and Torch. We're seeing less ice and for less period.
Okay, so you got to remember, I've thought about this for 6 years, and I've studied algae for a long time. So so that is a really interesting hypothesis. And the reason that I think it's a particularly interesting hypothesis is that there is a lot of GBA in the deeper water. And one of the things that I've studied a lot is dispersal of algae and the role of dispersal of algae in having algae grow someplace else. So imagine that with thinner ice cover and for a shorter amount of time, the GBA fundamentals, the GBA algae overwinter in the lake, and then as summer comes, because we do see more and more development in the shallow waters as the summer goes along. That seems to be a pretty strong pattern. That could be the algae from the deeper zones spreading to the shallower zones and facilitating the accumulation, feeding the accumulation in the shallow waters. And that we are studying that in the torch surveys now, where we're sampling shallow water and deeper water and looking at the synchronization of the deeper water GBA and the shallow water GBA. So that's really a very interesting hypothesis. It involves a couple steps in the mechanisms, and there's some other things that bug me about it. There's some other ecological connections that aren't made, but maybe if you think about it a little bit more, you make those connections. That's the whole idea about having that list of hypotheses and continually thinking about one versus the other and thinking about new observations. As frustrating as I know it is for you not to, after 8 years, not have found a solution to the problem.
Are there any larger lakes in Michigan that do not have GBA? And what about like the Bay of Lake Michigan? Those are also large in deep water.
So I sampled in Little Traverse, and I did not find GBA there. And actually, Lake Michigan, northern Lake Michigan, even has higher phosphorus. It's up around 10, or 8, 10, someplace in that range. If I remember the numbers, I don't remember the numbers exactly, but I remember them as being higher than, for example, where you really get this rigorous calcareous mat. Now, again, there are different kinds of algae and trying to relate these things, but the biggest mats are in Charlevoix, Torch, and Elk so far, the thickest mats. And there are our lowest phosphorus lakes. So no, I'm not getting it there. It's interesting, it's not in the Glen Lakes. So there's variability among lakes that should help us potentially understand that.
Question.
So can I take one from the chat here for just a second? Okay, here's one from the chat, and your mic's off, Bob. Did you mention that an increased nitrogen caused increased mat formation? Does decreased phosphorus follow increased nitrogen levels? So I didn't talk about this nitrogen deposition hypothesis much, and the idea behind that is there's work where people working with diatoms and doing paleoecology. I'm not sure how many of you are familiar with using sediment cores out of lakes to understand the past history of the lake. And So Sherry Fritz and John Kingston and Dan Engstrom did a study back in 1993 where they took cores out of many northern Michigan lakes, and they dated the cores so they knew how old the sediments were, and then they looked at proxies for environmental condition in the sediments at those in the cores. So going from recent sediments at the top to really old sediments at the bottom. And it turns out the proxy they used was the diatom species. The glass cell walls are preserved in the sediments. And diatoms are very sensitive to changes in phosphorus. So a shift from one group of species to another group of species can very precisely, precisely and accurately, closely, both these statistical terms apply, tell you what the phosphorus concentration is. So they used these methods and found that from about 1940 to the time that they sampled, the early 1900s, that it looked like there was a decrease in phosphorus in the lakes. In 4 of the lakes that they caught, and I think Bel Air was one, but they studied a lot of lakes. So anyway, what would cause that? Maybe nitrogen deposition might cause an ink, because it was around the 1940s that the Haber-Bosch process produced ammonia that was used as fertilizers all over, and then cars started emitting more nitrogen. And so nitrogen pollution was a really big problem that wasn't regulated until around 2000, which was about the time that Dreissenian mussels came in. So if the phosphorus was decreasing because of nitrogen, and then the Dreissenian mussels kicked in, that would explain this long decrease in phosphorus, this long-term decrease in phosphorus concentrations in the lakes. So why would nitrogen deposition cause that problem? If you add nitrogen to habitats, you can get shifts in species that are able to use that require that higher nitrogen. And those species might be able to use lower levels of phosphorus. And then they precipitate out of the water column, so your N phosphorus concentrations during the summer become lower over time as more and more phosphorus settles out of the water column, as you get nitrogen enrichment actually driving lower phosphorus concentrations in the lakes by algal uptake, by stimulating different species of algae that can take up phosphorus at lower concentrations. That was the idea behind the nitrogen deposition hypothesis.
Dan, you mentioned in the Florida experiments with phosphorus, and what I picked up on, you said the mats did not form. My question is, in the channels where the mats didn't form and phosphorus was fed, was there still the concentration of diatoms and the generation of the mucilage?
So we did not study the bottom algae very much, but it did stimulate algae on the bottom, and it caused a different kind of algae to grow, different species. So they also produced mucilages, but they produced mucilages in different ways that did not form tightly cohesive mats. They were more flocculent in nature. Does that answer your question? Did I hear it completely?
Yes.
Okay.
And I went for a lovely boat ride today on Elk Lake, and we probably covered 10 miles of shoreline. And the entire shoreline, out to 5, 6, 7 feet, was covered in this golden brown algae.
Yeah.
Getting home, I had to do some work, taking my dock out and whatnot, and I noticed that there was no algae under my dock. So where the sun wasn't penetrating, there was no algae. And I think I heard you say earlier that there was no relationship between the light penetration and the algae. My other observation is that I got out into deeper water, like 8 feet of water, the brown algae declined. So again, it's kind of inconsistent with what your comments were about this algae being in the deep water. So for a non-scientist, non-biologist, and not a very smart guy, it's a pretty simple correlation between what I saw as groundwater that had been penetrated by light versus, I should say groundwater, lake shore versus non-light penetration. Can you talk about my observations?
Yes. So I see GBA underdocks and out to deep water, out to 20, probably 15, 20 feet if the water's clear enough to see. Unless there's Cara, but I've actually got some videos of Elk Lake and Elk-Skegemog sampling and the GBA on the bottom if you'd like to see them. That show the GBA growing on the Cara, which is not always the case. But so I think, you know, we'd want a wider set of observations, of course, but I don't have a good light explanation. Of course, you have less light under your dock, but there might also be, might also be a shelter for animals to come in there and move around on the bottom and disturb the algae. That's a plausible hypothesis. One of the principal, the algae are really efficient at using light, especially benthic algae. That, and we, you know, I've done experiments on this in the past, actually up at the University of Michigan's biological station. And they will grow almost at maximum growth rates by shifting the amount of pigments in their cells with 1% of daylight. So, so they're very efficient at using low light. That said, your observation and that, you know, the idea of light potentially being a factor is a good one to consider. Around the shorelines too, so when I look at the aerial photographs, around the shorelines, wave disturbance is a huge factor in breaking up the algal mats. So I've actually started sampling much more at 6 to 8 feet, where wave disturbances shouldn't be as much of a factor. But I appreciate the observation and but yeah, and also you back it up. So has light changed enough in the lakes to have stimulated this growth to occur? Has seconded actually changed that much in the lakes? Seems to me it's actually increased, so you've gotten more light in the lakes.
Jan, we thank you for your presentation, and I think you will hear the noise from the audience. And I don't know that we've got that we know a clear way to eliminate the brown crud, but let me just ask you one more thing. What in 5 years do you expect that you'll know enough more to make a significant difference?
5 years? Well, I hope we know something in 5 years. But um, I mean, I wouldn't be working on it if I didn't hope that we would learn something in 5 years. But this is a huge problem, and there's not a lot of funding going into it. I mean, having USGS involved will really help. So that funding effort will, potentially link, what human activities and what's natural factors and how those might interact to affect water chemistry, and that's really important. So if we figure out the proximate causes of the GBA, and I think these experiments and the surveys are really going to help us figure that out. I hope we'll figure out what kind of management options are available to solve the problem.
Thank you so much.